All right, let's finish up the special senses here. So we're going to go back to vision just a little bit. There are a few things that we didn't get to cover the other day. The eye is organized into two segments, the anterior and posterior segment. In each of these segments, there is a humor, which is just kind of a fluid that exists in the eye. In the posterior segment, you have the vitreous humor. It's a bit more jelly-like, and it's clear, which is good because it needs to transmit light uh, through the eye. It also supports the back of the lens, the posterior surface of the lens in the eye, and then it holds the neural retina firmly up, like it holds it right up against the pigmented layer in the eye. It also contributes to intraocular pressure. If you ever go to the optometrist and there's that thing where they do the little puff, where I think it's usually like a farm in the distance that you're looking at, and then it puffs your eye, and it always startles me at least. That's measuring intraocular pressure, the pressure inside the eye. It's a really important thing for your optometrist to be able to figure out if it's too high or too low. In the anterior segment, you have the aqueous humor. The anterior segment it's made up of two smaller chambers, and they are confusingly named the anterior and posterior chamber. Don't get those confused with the anterior segment and the posterior segment of the eye. Those are two different things. And again, I cannot emphasize how much anatomists do not like students because they would name this. The anterior chamber, that's between the cornea and the iris. That's where the aqueous humor actually is. The aqueous humor is a much less jelly-like than the vitreous humor, the aqueous humor. You could see it's called aqueous as in water. It's water. It's more watery than the vitreous humor is. The posterior chamber of the anterior segment is the space between the iris and the lens. We'll see what we're talking about here in just a second. So you can see here, this whole bit right here, this is all the anterior segment. And then everything behind the lens would be considered the posterior segment. Okay. And then you can see this anterior segment here is separated into the anterior chamber, which here, this is where the aqueous humor is, right up to the cornea, all the way back to the lens and the iris. And then behind the iris, but in front of the lens here, that is the posterior chamber of the anterior segment. This is where you actually have that aqueous humor flows through down this sinus here, but don't worry about that. Just know that this is where the aqueous humor is in the anterior segment. I'm not especially worried if you know that this is the anterior chamber versus the posterior chamber. I just know that it says that here. I wanted to make sure that you saw that there is a difference there. The posterior segment here, that's where the vitreous humor is. In the eye lab, in the eye dissection lab, you'll see what I'm talking about with the difference between the vitreous humor and the aqueous humor, just in viscosity. But the posterior segment with the vitreous humor is behind the lens. Okay. All right. So that lens itself, we haven't really talked about too much. The lens is biconvex, which convex, just talking about angles. We'll take a look at that shape in a second. Bi meaning two. So it's biconvex. It's convex on two sides. It's transparent, which is super useful because light has to go through it. It's really flexible, and it's also avascular. Avascular, remember, means that it does not have any blood supply to it. All right. The lens is how you are actually able to focus light onto the retina, depending on what you're looking at. It's made up of lens epithelium and lens fibers. The lens epithelium actually differentiate into lens fibers, which are just these cells that are filled with a protein called crystalline. With age, as you get older, the lens, it gets more dense, it gets more compact. It's not quite as stretchy, which is mostly why you'll see older folks, they start losing their eyesight. That's because the lens just is much less flexible as you age. So you can see this is the lens right here, and biconvex means this is a convex side, and this is the other convex side. So as light comes through, I think we have a picture of representing the light coming through, and the lens focusing it. But it is actually really flexible. In the lab that we do, the eye dissection, you'll probably notice that the lens is not very flexible, but that's because the cow eye has been dead for some time. And when it's alive, this is actually fairly flexible. All right, and that's pulled on by the ciliary bodies here, and that's what stretches these out. Okay, so again, we're going to get into a little bit of physics here. So refraction, that's when light bends because it passes through different transparent mediums. So it's normally passing through air before it gets to your eye. Then once it gets to your eye, it passes through a few different transparent mediums, so it gets bent as it passes. So when it passes through a convex lens, it bends so that the rays converge onto a single point. All right. So you see, here's like where the light is coming from. The light is coming off from a few different angles here, and then they all converge as they go through the further away from the center they are. You see how it's pretty much a straight line if it's going diagonally like that. But if it comes all the way up here, it has a focal point so that the image is reconstituted at the end of it. All right. Through the convex lens, the image is actually upside down and backwards, like you can see here. As the light passes through, it goes all the way through into the other side here. The yellow box is down on this side, but then as it passes through, it's exactly flipped here. And that's actually what's projected onto the back of your retina, and your brain adapts to that. Right. So just to follow the pathway here of light as it goes through the eye. So you have light coming in. Here's the cornea. It enters the cornea. Then there's the aqueous humor. Then it goes through the pupil into the lens. Then it passes through the lens into the vitreous humor. And then it hits the neural layer of the retina, which we don't have blown up here, but this is the retina right here. Hits the neural layer of the retina, and then it gets to the photoreceptors. All right. That is 3 times that light is refracted. You have once at the cornea, once as it enters the lens, and once it leaves the lens into the vitreous humor. The way that this is curved and able to be the shape is able to be changed. That's how our eyes are able to focus on really fine details of an image. So when you're looking at something at a distance, the light doesn't really need a whole lot of adjustment in order to focus on it. Your lens actually only needs to change shape up to about 20 feet. After that, your lens doesn't really need to change a whole lot. That's because if you look at an object that's far away at 20 feet, it's the light that you need to have enter your eye in order to make up that image is pretty much parallel to the eye at this point. If it's much closer, then it's the angles are much sharper. But at this distance, the light that's coming into your eye is pretty much parallel in order to get focused on here. So it doesn't really need to change a whole lot once it's the lens is flattened out like that. At close vision, though, it's maybe a little bit counterintuitive. When the muscles contract, it bulges out, it rounds out the lens. That's accommodation so the lens shape can get the light that's at this, you see this little bit steeper angle where it's not parallel anymore. It's a steeper angle here. As it comes in, it needs to refract that so that you can actually focus on that. When this constricts, the pupil constricts so that there's not a whole bunch of extra light coming in off the sides here. You're just wanting to see this. So that's kind of how, when you're reading something, it takes a lot less, or it takes a lot more effort. And when your eyes get tired, you kind of look away, you look into the distance. This is why that is. So we have a few different issues with refraction. I'm sure plenty of you guys wear glasses or know someone that does. If your eye is emmetropic, that means that it functions as intended. It doesn't need to have any kind of adjustment in order for the light to focus properly. If you're looking at this right here, you see all the parallel beams come in, and they all focus exactly where it needs to be on the retina in order for the image to be recreated in your brain in focus. If you have myopia, or if you have a myopic eye, that means that you're nearsighted. That means that you need to wear a concave lens in order to be able to see. All right. This is what I have. My glasses are a concave lens. Nearsighted meaning that like you can see something that's close up. That's because the eye focuses all of this a bit in front of the actual retina here, where it needs to be. So by adding this concave lens, that just adds a little bit more, kind of spreads that out a little bit so that it projects that image so that it converges onto the actual retina, versus the hyperopic eye, which is farsighted. That means that it converges the image just behind the retina, and that uses a convex lens or a biconvex lens to refocus that right onto the retina. All right, we're going to get into gustation and olfaction now. There's just a couple of slides about this. So these are what we would call the chemical senses. It's gustation, which is taste. I mentioned the other day, I think of Gustave when, which I think of a French chef or something. When I hear gustation, that's how I remember that. If it helps you out to remember, like gustar in Spanish means to like. That actually does come from the same root there. That like when you taste something, you couldn't like it is the idea. But gustation is taste, and olfaction is smell. There's not an easy way to remember that. You just have to know that one. Or at least I don't know an easy way to remember that. With both gustation and olfaction, you have chemoreceptors, and those are the primary receptors in these senses here. The way that they work is they respond to chemicals in an aqueous solution, in a watery solution. With taste, those chemoreceptors respond to substances dissolved in saliva. And with smell, it relies on the fluids in your nasal membranes in order to dissolve those chemicals that come in for them to be stimulated. So with olfaction, you have the olfactory epithelium. That's the organ that actually functions here. This is not something that you need to have memorized, this image here, but this is hopefully for some kind of clarity as to what we're looking at, like with the olfactory bulb versus the olfactory cranial nerve. You have these bipolar cells here, right here. They have these olfactory cilia as they come down. Here, you see these olfactory cilia. These are the bipolar neurons. They have all these supporting cells around them, and they respond to a bunch of different kinds of odor-causing chemicals. All right, so you have the nasal passage here. Down here is where you have those receptor cells and the olfactory cilia. That's actually, the air goes by that, and different kinds of chemicals interact with the olfactory cilia, and that stimulates that and goes up through the olfactory tract into the brain, and that's where your brain processes smell. With gustation, you've got about 10,000 taste buds, and most of them are on the tongue. Right? They are found in the papillae of tongue mucosa, which papillae just means kind of like bumps or projections. The taste buds are found inside these papillae on the tongue. There are 3 different kinds. Only 2 contain taste buds. Don't worry about that. They're just, it has a couple of different kinds on here, and I just wanted to point that out. All right. You have 5 basic taste sensations that you can figure out. You have sweet. That's when the receptors are stimulated by sugars or saccharin or alcohol. There's some amino acids that register as sweet. Salty is another taste sensation that we have, and that's metal ions, which makes sense. You think of like sodium chloride, right? When metal ions stimulate the taste buds, then it tastes salty. Sour is hydrogen ions, right? That when a hydrogen ion stimulates one of them, that you register that as sour. Bitter tasting things are alkaloids, like quinine or nicotine. And then umami, which is maybe one that you're not familiar with. That's kind of like savory is the idea there. Like you think like beef broth or something like that. It's a savory kind of taste. That's glutamate is what stimulates that. Okay. So you see you have the taste buds here. These are where the ion or the molecule enters. And this is the receptor cell. It registers here, and then it goes up through the cranial nerve. The signal gets sent through the cranial nerve to the brain, and that's how you process your sense of taste. So it is mostly taste is like 80% smell. That's how your brain registers that. There are also a few other things that affect taste. You have thermoreceptors, mechanoreceptors, and nociceptors. Just how you're able to, and those all influence taste. Uh, like, you know, if something is hot, then it can taste different than if it's cold. And texture also can impact that, which is where those mechanoreceptors and the nociceptors come into play. All right. I believe that is all for the rest of the special senses.